Hello, everyone. Thank you for taking the time out of your days and joining me for today's presentation. My name is Stephen Diaz. I am a student currently studying at Stanford University and working with the Sharaf lab here on campus. Today, we will be exploring the thermal stability of a lipoprotein nanoparticle. Nanoparticles have emerged as some pretty promising tools for vaccine and therapeutic design. The Sharaf lab has developed a novel biomaterial based on bacterial lipoproteins. We call this material the lipocatch. This material self-assembles into a nanoparticle under proper conditions. Because this biomaterial is so new, a thorough characterization of its properties is sort of lacking. My project uses multiple biophysical tools to characterize the thermal stability of lipocatch. This work, I hope, will aid in the design of novel immunotherapies. So let's get talking on my first step here. So my first step was to produce the nanoparticle. To produce lipocatch, I used recombinant DNA technology to overexpress a bacterial lipoprotein in an E. coli expression system. A whole cell STS analysis displays a band right here at the theoretical molecular weight of our protein, suggesting that our protein was indeed overexpressed and thereby present in solution. I then purified my bacterial lipoprotein using affinity followed by column size exclusion chromatography. A representative graph of the chromatography is displayed right here, with the x-axis representing the elution volume and the y-axis measuring absorption at a 280 nanometer wavelength. An SDS page analysis of the purified solution shows that my protein was present in both the first and second peaks of the chromatogram. This suggests that there is some sort of equilibrium, as we attempted to display right here, between a monomeric and self-assembled nanoparticle in our purified solution. Moving to methods. Method 1. My next step was to characterize the thermal stability of lipocatch using the nano-DSF technique. Nano-DSF is based on the intrinsic fluorescence of tryptophan, an amino acid that is sensitive to its local chemical environment and known to be present in our lipoprotein. A representative graph is shown here. On the x-axis is the temperature, and the y-axis is the 350 to 330 nanometer fluorescence ratio. As temperature increases, we detect a change in the fluorescence ratio with an inflection point of around 49 degrees Celsius. This suggests that the shape of our sample composition is influenced by temperature, a suggestion that we will be further exploring further down in this poster. To look into this composition alteration in further detail, I use the dynamic light scattering technique, or DLS for short. DLS is an in-solution experiment where a laser is directed at a protein sample and the scattering of light, represented over here, is collected and used to make inferences on the size of our particles. Big particles, as represented over here, diffuse slower and exhibit slower intensity fluctuations in comparison with our smaller particles due to the laws of Brownian motion. An autocorrelation function, represented in letter D over here, is then created measuring the extent of particle movement in solution. Smaller particles exhibit faster decay due to their faster movement and diffusion within the solution. After that, a theoretical model is built based on a predetermined globular protein structure of defined size and a comparison of the theoretical model and experimental autocorrelation function give an estimated hydrodynamic radius. And so here we estimate that the smaller particle would have a smaller radius, And then as defined in the purple, hopefully that would have a bigger radius. Moving towards our results, they are displayed right here. Here are my DLS results. In this experiment, 5 DLS measurements are required at intervals of 5 degrees Celsius. So we move up 5 degrees and we collect 5 acquisitions per each temperature. Representative DLS traces for 3 select temperatures, 25 degrees, 60 degrees, and 75 degrees, are shown here 1, 2, and 3. On each graph, the y-axis represents the relative intensity percentage, and the x-axis represents the hydrodynamic radius measured in nanometers. At room temperature, otherwise known as 25 degrees Celsius, we detect one peak at approximately 10 nanometers. At 60 degrees, we detect 2 peaks, and at 75 degrees, we almost exclusively detect only the second peak. I then analyze this data to create the graph below, depicted over here. As you can see, as temperature increases, the fraction of peak 2 detected also increases, as peak 1, represented in the green, decreases. Based on the DLS results, the fraction of peak 1 versus peak 2 measured undergoes a major transition at around 55 to 60 degrees Celsius. Together, the nano-DSF data and the DLS data suggest that near body temperature, human body temperature, which is around 37 degrees Celsius, the sample composition remains relatively stable. So we'll be over here and we see, peak 1 represents most of our protein in solution. For some concluding statements, I successfully expressed and purified the lipocatch protein up here. I used a buffer exchange technique to create the lipocatch nanoparticle. The thermal stability of lipocatch was then measured via 2 separate biophysical tools. And the results indicate stability well past average human bodily temperature, prompting further questions and experiments regarding in vivo usage of our nanoparticle. In the future, we plan to load the nanoparticle with small molecule model compounds and determine the change in particle distribution as functions of temperature. I'm really looking forward to seeing how this project progresses. Thank you so much for listening, and I hope to see you all again soon. Thank you so much.